The study of biology encompasses a large number of different subdisciplines that range from biochemistry to ecology. In all of these, however, there are unifying themes. Among these are the cell theory, the molecular basis of inheritance, the relationship between structure and function, evolution, and the emergence of novel properties. As we stated at the beginning of this chapter, all organisms are composed of cells. This is life's basic unit, and all cells come from pre-existing cells. Cells were first observed in 1665 when Robert Hooke used one of the first microscopes that magnified them 30 times. Not long after that, there was a Dutch scientist named Leeuwenhoek who used the microscope that was capable of magnifying things 300 times. These three concepts constitute the cell theory. The molecular basis of inheritance explains the continuity of life. Even the simplest cell is incredibly complex, and the information that's specified by each cell is encoded in DNA, or deoxyribonucleic acid. This is a long cable-like molecule. You've probably seen one in a picture somewhere. We'll investigate it in much more detail later in the semester. It's pretty amazing to think that just a sequence of 4 different letters, or nucleotides, A, T, C, and G, encode the whole cell's information in all of the trillions of cells in our body. On the DNA, we'll find genes, which are discrete units of information. Not all the DNA contains genes. They're just scattered throughout each of the chromosomes. The entire set of DNA for any individual, all the instructions for that individual, is called the genome. And the continuity of life depends on the faithful copying of DNA into daughter cells. We'll investigate this process later on in the semester. It may seem fairly obvious that structure and function are related. We can study the structure to learn the function of a particular enzyme, or maybe even a beak shape. And if we know the function of a protein, we can look for the same structure in other organisms. For example, if we know the receptor on a human cell for insulin and its shape, and we find a similar molecule in the cell membranes on a worm, we might be able to conclude that this molecule functions in a similar way and establish some evolutionary relationships between glucose utilization in worms and humans. Another unifying theme is that diversity of life arises by evolution. The underlying unity of biochemistry and genetics argues for life from the same origin and event. We address this in detail in a chapter later on in the full textbook, and that we see diversity due to evolutionary change over time, as exemplified by Darwin's finches. The diversity of life has brought us 3 different domains. Now, you may be familiar with the old system of simply 5 kingdoms: plants, fungi, animals, protists, and then a group we used to call the monerants. We've now recognized that they are much different than any of the organisms involved here: plants, fungi, animal, and protists. And so we've created one higher level called the domain level. The bacteria are single-celled prokaryotic cells. And the archaea are also single-celled prokaryotic cells. However, these are the extremists. They like very extreme, acidic, hot, or sulfury type of environments. And they're actually more similar to the eukaryotes than they are to bacteria, but that's a whole nother story. The eukarya, however, are single-celled or multicellular eukaryotes. The protists represent all of the single-celled eukaryotic organisms, and then plantae, fungi, and animalia represent all of the multicellular eukaryotes. As you'll learn early in the semester, prokaryotic cells and eukaryotic cells are distinctly different from each other. We'll start by just saying that eukaryotic cells are about 10 times larger. So all the organisms of the domain Eukarya have large cells versus the smaller cells that we see in the domains Archaea and bacteria. Biologists agree that all organisms alive today have descended from some simple cellular creature that arose about 3.5 or 3.6 billion years ago. Some of the characteristics of that earliest organism have been preserved. The storage of hereditary information in DNA, for example, is common to all living things. The retention of these conserved characteristics in a long line of descent usually reflects that they have a fundamental role in the biology of the organism, one not easily changed once adopted. A good example is provided by the homeodomain proteins, which play a critical role in early development in eukaryotes. These conserved characteristics can be seen in approximately 1,850 homeodomain proteins distributed among 3 different kingdoms of organisms. Here's just a few examples of different homeodomain proteins that we see in plants and mammals and fungi. Here's a different family, also found in plants, animals, and fungi. Another unifying theme is that cells are information processing systems. Information in the DNA is used to direct the synthesis of cellular components, and the control of gene expression leads to different cells producing different tissue types. For example, although every single gene is in our hair cells, as well as in the cells we might find in our eyeballs, not all of the genes are expressed. Eye color is turned on in the cells in our eyeballs, whereas hair color is turned on in the cells of our hair. Wouldn't it be interesting if that got reversed somehow? Cells also process environmental information. For example, we measure glucose levels in the blood and the presence of hormones in the blood. Each cell has receptors for different substances, which are controlled by all those things that control gene expression. Cells in multicellular organisms also must coordinate with each other so that the system runs smoothly. Now, it's important to note one of the key features of living systems is that they are open systems that function far from the thermodynamic equilibrium. This has a number of implications for their behavior. A constant supply of energy is necessary to maintain a stable non-equilibrium state. These self-organizing properties of living systems show up at different levels in the hierarchical organization. At the cellular level, we see macromolecular complexes, such as the spindles that are necessary for chromosome separation and mitosis. And at the population level, we see schools of fish and flocks of birds or bacteria on a biofilm also self-organize. Finally, there are emergent properties that we see from collections of molecules, from collections of cells, and from collections of individuals. You couldn't predict how dogs would behave in a pack by simply looking at one dog, just as you could not predict how all the cells of your eyeball just by looking at one of those cells. So that brings us to the end of our first chapter of the semester. Now it's time to go online. First of all, I recommend doing the Learn Smart exercise through Connect. And then you can take your chapter quiz because you'll have a great understanding of the material by then. In addition, there'll be a diagramming assignment for each of the chapters. So you want to make sure that you take care of that and any other things listed under this chapter.